Sometime in the next 24 hours, you're going to be alone in a room with something that makes you feel better, and you're going to call it relaxing. In 1977, on a university campus in British Columbia, a psychologist built a small paradise for rats to find out what that moment actually is. He gave them everything a rat could want. Then he offered them morphine, sweetened like candy. What they did next is still the most inconvenient result in the history of addiction science. Because if he was right, the thing you've been calling a weakness in yourself was never in you at all, and it starts with a cage the size of a shoebox. The psychologist is Bruce K. Alexander, and in 1977 he is teaching at Simon Fraser University in Burnaby, British Columbia. At that moment, everything science knows about addiction comes from an animal in a box. A standard laboratory rat cage gives one rat about the floor space of a shoebox, alone, under fluorescent light, for its entire life. In 1969, Gerald Deneau and his colleagues gave monkeys a vein line and a lever wired to cocaine. The monkeys pressed until they died. That result becomes the whole story. The drug is a hook. Flesh is flesh. Anyone who touches it goes down. So picture yourself in that cage. Bare wire, no company, nothing to do, and two bottles. One is water, one is sweet and takes the edges off the day. So, do you take the drink? Hold that answer. You'll need it in about 15 minutes. Alexander is a bearded professor who has spent years staring at those cages and asking a question nobody in the field is asking. Not what does the drug do to the rat? What does the cage do to the rat? So in 1977, with Robert Coams and Patricia Hathaway, he stops studying rats and starts building real estate. They take plywood and build a box 8.8 square meters across, about 95 square feet. That's about 200 times the floor area of the cage the rat was living in before. They fill it with cedar shavings, tin cans, running wheels, and small wooden boxes to hide in. They put 16 to 20 rats inside, males and females together, and let them do what rats do, which is everything at once. And then, on the plywood wall, somebody paints a mural. A British Columbia forest, trees, a stream, hills in the distance, painted for animals that cannot see it properly. Nobody made them do that. The lab calls it Rat Park. And here is the part nobody tells you when they retell this story. Alexander is not testing morphine. Morphine is the boring variable. He is testing whether the last 50 years of addiction research measured a drug or measured a cage. If he's right, the entire field spent half a century studying loneliness and writing the word heroin on the results. So there's only one thing left to do: offer the drug to animals that have something better to do. The first problem is that rats hate morphine because you can't test a drug on an animal that won't touch it. Raw morphine hydrochloride in water is bitter, and a rat with any other option will refuse it. So Alexander's team does something that should be in a museum. They add sugar, then more sugar, then more, until the bitterness disappears and the rats will drink it. It's a documented laboratory technique called sucrose fading, and what it means in practice is this. In order to prove that rats will get hooked on opiates, science first had to turn the opiate into a milkshake. Both groups get 2 bottles, plain tap water on one side, sweetened morphine on the other, unlimited day and night for weeks. The caged rats drink the morphine. The Rat Park rats mostly don't. In the strongest conditions, the isolated rats consume up to 16 times more morphine than the animals in the colony. Same species, same litters, same drug, same sugar. The only difference in the whole experiment is the room. The 1978 paper is titled around 2 variables, housing and gender, and the second one surprises them. The isolated females drink the most morphine of any group in the study. Nobody at the time has a clean explanation. It's simply in the data, sitting there. It gets cited far less often than the headline number, which tells you something about how findings travel. Now, if you're skeptical, you've already found the hole. Maybe the Rat Park rats just don't like the taste. Maybe they're too busy to bother walking to the bottle. Alexander's team thought of that, and their answer is the most elegant thing in the entire study. They add naltrexone to the sweet solution. Naltrexone blocks the opiate effect and leaves the sugar. The Rat Park rats drink it happily. They wanted the sugar the whole time. What they were avoiding was the high. Let that land. The happy rats weren't refusing a flavor. They were refusing to be sedated. Alexander is standing at the plywood wall watching this, bearded in front of a forest that somebody in his lab painted by hand. But a critic can still say the obvious thing. Those rats were never addicted in the first place, so the team runs the cruelest version of the experiment. For 57 days, morphine solution is the only fluid available. Every rat, both housing conditions, no choice at all. Drink it or dehydrate. By the end, every animal in the study is physically dependent, not psychologically, chemically. Then they open the second bottle and put plain water back in the room. The caged rats keep drinking morphine. The rat park rats walk away from it. They choose water while they are still in withdrawal. You can see it in the recorded observations. The shaking, the twitching, the wet dog shudder that rats do when opiates leave the body. They go through it anyway because there is a wheel and a tin can and another rat and the day is busy. That is the finding. Physical dependence is real, and it is not enough. Every treatment programmed in this country was built on the opposite assumption. The chemistry hooks the body. Something else decides whether the body cares, and there's a second version of that finding that should be printed on a wall somewhere. In the 1981 follow-up, the team took rats that had spent their whole early lives isolated in cages and moved them into the colony. A miserable childhood, then a decent room. They didn't drink much morphine either. Your history loads the gun. Your present decides whether anyone picks it up. Here's the part the retellings skip. Alexander writes it up and sends it to Science and to Nature, the two journals that decide what counts as knowledge. Both rejected. The paper lands in 1978 in Psychopharmacology, a good journal that almost nobody outside the specialty reads. The follow-up lands in 1981 in Pharmacology, Biochemistry, and Behavior. And then nothing happens. No press cycle, no policy change, no textbook rewrite. Meanwhile, the shoebox cage studies keep running, keep getting funded, and keep producing the number everyone already believed. In 1985, Michael Bozarth and Roy Wise publish a study where rats with unlimited intravenous cocaine die at a rate of 90% within 30 days. That result goes everywhere. The rats in that study were housed alone. In 1981, Simon Fraser University stops funding Rat Park. The plywood comes down, and Alexander is left holding a result that two journals have already told him nobody wants, which raises a question he cannot answer from a lab in Burnaby. Do human beings work the same way? In 1971, 6 years before Rat Park, the United States has a problem it cannot say out loud. American soldiers in Vietnam are using heroin, and the heroin there is close to pure and cheap enough to be an afterthought. Roughly 20% of enlisted men report being addicted to it, and the war is ending, which means all of them are about to fly home. Washington panics. In June 1971, Nixon stood in front of the press and called drug abuse public enemy number one. That same month, he creates a new White House drug office and puts a psychiatrist named Jerome Jaffe in charge of it. The military starts urine testing soldiers before they're allowed to board the flight home. The official nickname for that program, which somebody in a uniform wrote down and approved, is Operation Golden Flow. And then the government accidentally runs Alexander's experiment on returning soldiers and gets an answer so unwelcome that the people who commissioned it refuse to believe it. Lee Robbins is an epidemiologist at Washington University in St. Louis, and she is given something no researcher gets: access to the returning soldiers, their urine tests, and their Army records. She tracks a sample of over 900 enlisted men who came home in September 1971, and she interviews them again years later. Everything known about heroin says these men are finished. In the 1970s, the accepted relapse rate for treated heroin addicts is somewhere above 90%. 90% after treatment, after detox, after everything. Robbins finds that of the men who were addicted in Vietnam, about 5% relapse in the first 10 months after coming home. 3 years out, it's about 12%, which means roughly 9 out of 10 men addicted to pure heroin in a war zone came home and simply stopped. Sit with that gap. 90% relapse in the clinic, 5% among the men who flew home. Same drug, different room. No willpower seminar, no 12 steps. They got off a plane into a country where they had a mother, a girlfriend, a job, a truck, a street they knew, and the cage was gone. This is the moment the whole story could have turned. 2 independent results, one in rats and one in returning soldiers, pointing at the same conclusion, 4 years apart. Robbins published the findings in 1974, and the reaction was not relief, it was disbelief. Robbins spends years afterwards explaining that her data are real. The field's response is to decide that Vietnam was special. The heat, the war, the unusual purity of the supply, anything except the obvious sentence, which is that people use drugs to survive a situation and stop when the situation stops. Nixon's sentence became 50 years of policy, and it rests on an assumption nobody checked. It assumes the enemy is the chemical, and in fairness, the science is not clean. Rat Park has never been a slam dunk, and pretending otherwise is how good ideas get killed. In 1989, Michael Bozarth, Anthony Murray, and Roy Wise ran a version using intravenous heroin instead of a drinking bottle, and group-housed rats took more of it, not less. Other labs got partial effects, or effects that depended on how the animals were raised. The honest summary is that housing changes drug consumption, and how much depends on the drug, the root, and the rat. That's a smaller claim than the internet makes. It is also, unmistakably, not nothing, because the smaller claim is still the one the last 50 years of policy ignored. If the environment sets the ceiling on how much a body wants a drug, then a policy that isolates people further will raise the ceiling. We built that policy. We arrested people, took their jobs, took their housing, and separated them from everyone they knew. We took the rat out of the colony and put it in the shoebox, and then we measured how much it used and called the number proof. In 2021, more than 100,000 people in the United States died of a drug overdose. It is the largest number in the country's recorded history. Most of them were not in a laboratory, and most of them were alone. Alexander's argument is not that drugs are harmless, it is that we have been treating the symptom and protecting the cause. So what is the cause, in his own words? Alexander calls it dislocation. In his 2008 book, The Globalization of Addiction, he argues that modern societies produce addiction the way a factory produces a byproduct. He points out that addiction rates explode in populations that have been displaced again and again across centuries, not because the drugs got stronger, but because the belonging got weaker. Communities break. Work stops meaning anything. People move for jobs and lose everyone they know. And then something arrives that makes the afternoon bearable, which leaves one question, and it isn't about rats. What exactly is your cage? Here's where it gets uncomfortable, because a cage doesn't have to look like one. A cage is any environment that removes the things a creature would otherwise be doing. The rats in the shoebox weren't tortured. They were fed, watered, warm, and safe. Safe and empty is the whole recipe. And Alexander, bearded, standing in a lab in Burnaby in 1977, was pointing at a painted forest on plywood and saying, "It doesn't take much, a wheel, a tin can, something to climb, another body in the room." That was the entire intervention. Not abstinence, not shame, not a lecture about dopamine. Somewhere to be and someone to be there with. That's not a slogan. That's the experimental condition. And when a country has tried that at scale, the numbers move. In 2001, Portugal decriminalized personal possession of every drug, including heroin, and spent the money on treatment, housing, and getting people back into work. 20 years on, drug-related deaths in Portugal are among the lowest in Western Europe, and new cases of HIV among people who inject drugs collapsed from over 1,000 a year to a few dozen. They did not make the drugs weaker. They made the cage bigger. The critics of that policy are real, and they argue about the numbers to this day. But nobody argues the deaths went up. In 2015, a journalist named Johan Hari put Rat Park in a book called Chasing the Scream, and 38 years after the plywood came down, the study finally became famous. By then, Alexander had been retired from Simon Fraser University for a decade. Hari compressed the whole thing into one line, and that line is why it spread. The opposite of addiction is not sobriety, it's connection. But the sentence underneath it, the one from the plywood box, is harder. Alexander never cured a single addicted rat. He just took the cage away, and the addiction left with it. Everything after that is arithmetic. If the cage is the variable, then the question stops being what's wrong with the addict. It becomes, what did we take out of the room? And that question does not stay politely inside the topic of drugs, because you are almost certainly not in a cage that anyone locked. You're in one you assembled, one convenience at a time. Groceries arrive. Work happens in the same room where you sleep. Every friendship has a version that requires no travel and no clothing. In May 2023, the United States Surgeon General, Vivek Murthy, issued a formal public health advisory about loneliness. In it, between 2003 and 2020, the time an average American spent in person with friends fell from about 60 minutes a day to 20. Two-thirds of it gone inside 17 years. The advisory puts the mortality risk of that isolation in the same range as smoking up to 15 cigarettes a day. So think about this week, the night you canceled on somebody, ordered food, and stayed in with a screen, and told yourself you needed the rest. You weren't resting. You were in a shoebox with a bottle that takes the edges off, and the reason you never call it a cage is that somebody painted a forest on the wall. Nothing in that room is a punishment. Everything in it is comfortable. Your feed is the sweet water. Your apartment is the plywood, and you chose all of it, which is exactly what makes it hard to see. So let me ask you the question from the beginning again. So, do you take the drink? You already know the honest answer, and it was never about how strong you are. It was about what else was in the room. If that landed, subscribe. This channel is a list of experiments that quietly explain your own behavior to you. And tell me in the comments what your tin can is, the thing that makes the room worth being in. Then go put something in the room, a wheel, a person, anything. You're not broken. You're alone in a very nice cage.